Hello, everyone, and welcome back. This is Lecture 2, but we'll be wrapping up Chapter 1 and then continue on with Chapter 2 in a moment here. Before we begin, let's cover some learning objectives. With a wrap up of Chapter 1, we will be able to identify the significant and critical contributions to microbiology, compare biogenesis versus spontaneous generation, and describe phylogenetics, the three domains of life, and some important groups within each domain. How we see the world long ago, the predominant thought was that diseases were caused by supernatural forces. In 36 BC, Marcus Terentius Varro was the first person to suggest on record that living things that we cannot see may cause disease. In 1665, Robert Hooke developed a powerful microscope that allowed him to observe living organisms and documented the observation of, as well as coining the term cell. By 1675 emerges Anthony van Leeuwenhoek, who was able to polish lenses to a high degree and allowed him to observe microbes that he called wee little beasties for the first time in rainwater. Along the same timeline, in 1668, a man by the name of Francesco Redi was the first to provide evidence against a term called spontaneous generation. We will cover spontaneous generation in more detail in the future. First, I was able to find the quote from Marcus Varro in 36 BC, who postulated living things cannot be seen with the unaided eye that cause disease. In his book, it reads, "Precautions must also be taken in the neighborhood of swamps, both for the reasons given and because they are bred certain minute creatures which cannot be seen by the eyes, which float in the air and enter the body through the mouth and nose, and there cause serious diseases." "What can I do," asked Fundanius, "to prevent diseases if I should inherit a farm of that kind?" "Even I can answer that question," replied Agrius. "Sell it for the highest cash price, or if you can't sell it, abandon it." Now, we don't know where Marcus Varro got these concepts from, but it is interesting to note that the characters in his book were so confident about the existence of microbes, their causing of diseases, and their transmission, that they were willing to tell Fundanius that if he inherited a farm in a swamp, that it would be better to abandon it than to keep it. About the same time that Robert Hooke and Antony van Leeuwenhoek were examining microbes, there were two competing hypotheses about the creation of living organisms on Earth. Those two hypotheses were biogenesis and spontaneous generation. Spontaneous generation was the older of the two. It was the hypothesis that living organisms can be generated from non living matter, and this was a consequence of observation of inanimate organic compounds and the creation of life from them. For example, if people left meat out, eventually they might have maggots that appear on the meat. And this appears to support that living organisms can generate from non living matter. This is how people explained the appearance of these maggots. The second hypothesis, biogenesis, assumed that all living organisms are derived from other living organisms. Francesco Redi had one of the most famous experiments to test these hypotheses. Francesco Redi believed that biogenesis was the answer to how living organisms are created on Earth. And so he set up this experimental design to test it, including 6 jars, basically 3 different sets. Shown on the bottom was the first set, where a set of jars had a piece of meat put inside of them, and they were uncovered so flies could fly into the jars, lay their eggs on the meat, which were microscopic eggs and weren't visible to us, but their maggots would then appear. And this would make it appear to many that spontaneous generation had occurred because there was no direct link between the flies, the eggs, and the maggots. In the second set of jars in the middle there, Francesco Redi put a piece of gauze over the top. So the flies would lay down on the gauze, but they wouldn't be able to get to the meat. And And therefore, because they smelt the meat beneath it, they would lay their eggs there, and the eggs would then hatch, and you would get maggots on the surface of the gauze. And then the third set of jars on the top there, you put a cork on the jar. And naturally, with the cork there, the flies couldn't smell the meat. They wouldn't lay their eggs on the jar lids either. And so you had no flies and you had no maggots. In this way, Francesco Redi demonstrated that the maggots came from the flies who were laying their eggs on the meat, and therefore he lended evidence against the concept of spontaneous generation. This was very cleverly designed in its experimental setup. As we can see, we have a positive control. That is, the jars without the lids should have a positive result, the production of maggots. In other words, they should have maggots growing on the meat, and that's what we're trying to measure here. He also has a great negative control with the jars with the corks in place there. There should be no maggots there as a result because there's no flies allowed in. And then there's the middle set of jars with a gauze and the experimental sample set that allows air to go in and out. What was the reason the gauze was so important to add instead of just stopping the research with a set of jars with the corks? It was important because for a while scientists and individuals said that what allowed spontaneous generation to occur was something in the air. It was some magical essence in the air that allowed spontaneous generation to occur. So, Reddy cleverly set up the gauze, which allowed air exchange, but it did not allow flies to be exchanged. However, this wasn't good enough for those critical of biogenesis, and even more work was going to be needed to persuade them, which we'll see later. This just goes to demonstrate how rigorous science needs to be in order to go from a hypothesis to a theory. The reason why this wasn't good enough. This might apply to the specific case of flies, but what about microbes? Remember, this took place around Robert Hooke's time when he shook the scientific world up with his work on the existence of microorganisms. And people noticed that if you leave food out, like meat, you also have microbes starting to take over that meat as well. So people who were in the spontaneous generation camp weren't all that moved by this experiment. Lazzaro Spallanzani got to work on the microbial testing. He set up a similar experiment to Reddy, but with broth. He boiled the broth to kill all microbes and left one to the open air and corked the other. Only the uncorked broth grew microbes. The tricky part was that he couldn't use a gauze because the microbes were small enough that they could slip through the mesh and contaminate the broth. So, a man you're probably familiar with, at least by his name, Louis Pasteur, took up the challenge 200 years after Francesco Redi's original work. Louis Pasteur definitely disproved spontaneous generation, put the final nail in the coffin there with his famous swan necked flask experiment. And this is how the experiment was conducted. He followed Lazzaro's experimentation protocol. However, he did a little trick to the flask itself. He made what we call a swan neck in the flask. He would take the neck of the flask and he would heat it in flame, and then he took a pair of pliers and he stretched the neck out. And if you notice, it kind of has an S curve in it where it goes up and then down and then back up again. And then he would boil the broth for, I believe, about an hour. He would then observe that after letting it sit out, no contamination occurred in that flask. Now, if he did the same thing, he brought a flask up to boiling, then he let it sit out, no contamination occurred, and it was fine. But when he broke the neck off, a spoilage would occur. And feel free to pause the video and try to figure out why this was the case uh, with the broken swan neck flask, but not the unbroken one. Well, technically, the textbook is incorrect here. Basically, it says that the swan neck flask prevented air from entering the tube. But really, the key here in these experiments was not preventing air by putting a trapped amount of water here. Really, the key was allowing air to go through. And so, unfortunately, this error is in the textbook. But um, in the as you can recall in the previous experiments, the issue with the spontaneous generation people was that you weren't allowing the magical essence of air to have access. What he was able to do here was not prevent microbes from getting inside of the flask and allow free exchange of air. In this process of going through these curves, the microbes would get deposited in the basin of the curves there. So let me get my laser point out and see right here, there's no water here, but really, or broth, but really the microbes get trapped along here because barring any strong turbulent air forces, which are prevented by this long, narrow tube here, um, just like everything else on planet Earth, these microbes are governed by gravity. So they will fall to the bottom and also stick to the sides of the glass. And we still have his swan neck flasks in museums today that are still sterile, which is a testament to this experiment, a living testament demonstrating that spontaneous generation was a false hypothesis. And by the way, Louis Pasteur went on to invent pasteurization and make contributions to beer and wine brewing, as well as the germ theory of disease. And here we can see one of his flasks still on display today at the George Marshall Medical Museum. And you can see the broth there at the bottom, and there's no fluid trap in the bottom of that neck of the flask. And you'll note that it's a very clear, syrupy broth there, and there are no microbes growing in that material whatsoever. So that was about 1861. Now we've covered briefly the development of observing microbes, the development of demonstrating biogenesis and disproving spontaneous generation, and a little bit about the scientific methods and the rigorous science that had to go behind that now. And now in parallel, let's talk a little bit about the germ theory of disease. Let's begin the conversation with Ignaz Semmelweis in 1847. He demonstrated the support for what we call the germ theory of disease. That is, that some diseases are caused by germs. Uh, this basically boils down to hand washing practices. What he observed was that women who were treated by midwives as opposed to doctors while giving birth were much less likely to contract infections and die. Why would this be? See, he recorded that doctors who performed autopsies on the dead would then immediately go and assist with births, and this had a higher rate of infections and death with their patients. Therefore, he proposed that washing hands with chlorinated lime water in between patients would reduce the transmission rate of disease. In 1867, Joseph Lister, you might know him for the dental brand name named after him called Listerine. Joseph Lister implemented early aseptic techniques. What is aseptic technique? These are methods of working in order to be without or to avoid sepsis. Remember, A means without in patients. And he further supported the germ theory of disease. So he picked up on Ignaz Semmelweis's tips about hand washing, and he insisted that surgeons use extreme cleanliness and wash with phenol spray during surgery. And we'll cover why phenol is an effective antiseptic later on. At this time, high rates of surgical patients were dying from post op infections. At the time, his strict methods were extremely controversial, and many doctors fought tooth and nail against Joseph Lister because what he was basically saying was that doctors were killing their own patients. Thankfully, eventually, scientific reasoning won out on this one, and many, many people's lives were saved during the American Civil War. Moving along in 1876, Robert Koch discovered that the bacterium Bacillus anthracis was the cause of anthrax. And this adds again more to the germ theory of disease. He did so by isolating it from the blood of multiple infected cattle. And in 1928, Alexander Fleming had a wonderful discovery. He went on vacation actually and came back to his lab, and his lab was a little messy, and he had some old plates. Plates are just a solid medium, kind of like jello, which you can grow microbes on top of. And he had some staphylococci sitting out in this lab, which is a type of bacterium. And while he was cleaning up, he observed that there was a mold contamination growing on one of his plates, kind of like what you can see in the bottom left hand photo here. This is a mold outcropping here forming up. And this streak here is a bacterial culture that's been wiped along the plate and allowed to grow up on this plate. So you can see in this here in this bottom left hand photo, an image very similar to what he probably saw. And what was fascinating about this mold, which was named Penicillium notatum, was that around the mold, if you look at this mold here, there is no growth of bacterium. See this halo where no bacterium grew around this mold? Well, why is that? Well, the streak is the bacterium growth on the plate, but the bacterium was unable to grow in close proximity to the mold. And um, we'll apply this principle in our lab where we analyze the effectiveness of antibiotics. So this does have an application later on, we'll see. But Alexander Fleming essentially discovered that there's a chemical that mold produces that repels or has antibacterial properties. And that chemical is called penicillin. And this was one of our first antibiotics in modern science that we are all familiar with. And many of our current antibiotics are some sort of derivative of penicillin. We've come a long way since then, and now we have developed aseptic techniques, we have food handling and sterilization, we have our vaccines against diseases caused by germs, we have antibiotics, antivirals, and antifungals. And also, along with that, we have some new challenges such as antimicrobial resistance. We also have microbial biologics, or microbial biologics meaning drugs that are created from microbes themselves in our ever expanding bio based economy we covered in the last lecture. And no discussion of the history of microbiology would be complete without also discussing the modern phylogenetics. Taxonomy in biology is a science of naming, defining, and classifying groups of biological organisms on the basis of shared characteristics. I think we're all pretty familiar with that. Some of us have heard of the five kingdoms, which is now an outdated model that will be relegated to the dustbin of scientific history, just like abio or spontaneous generation, except for all but some outdated high school textbooks, as a classification system of referring to different biological organisms by breaking them down into groups based upon their traits, how similar they look to each other. However, in the 1970s, a scientist named Carl Woese in the United States developed a much more refined method of classification by studying genetics to determine the evolutionary relationship between organisms. He sequenced the genes of a piece of a relative to the DNA called ribosomal RNA. We'll get into what ribosomal RNA is in the future when we talk about genetics. But he chose this RNA, which is a type of genetic material, for two main reasons. The first is they are an agent protein, ribosomes, that are present in all living organisms. Being common in all organisms is very handy. It allows all organisms to come under its same classification system. Ribosomal RNA are required by all life forms, so they're present in all organisms and they trace back very, very far into the lineage. The second thing is that these ribosomes have been around for so long that they are pretty well optimized and therefore don't go through much evolutionary change over time. And so, therefore, they're what scientists call highly conserved genetic sequences. That is to say that their sequences change very slowly over time. And the little differences between one organism's ribosomal RNA sequence and another's will tell us how closely related each organism is to one another. In a way, it's as if you were to take a book that has been around for a long time, like the Holy Bible, and you were able to look at the changes from copying it and determining in order which one is older, which one is newer, and which copies were made from other copies, and which one shared a copy long ago. And we could develop something that would look like this with a book with different versions of it. And that is because if you have an error made from writing this ribosomal RNA code or this book of life from one individual to its offspring, then that offspring should also have that same error in its code written down as well. And keep in mind, we are all mutants, each of us as individuals. Every time one copy is made or one new person is created into a new copy for the next generation, there's an error rate of about 3 mutations per generation. So all of us have about 3 different mutations or misreadings of our code in the previous generation's book or our parents. What Carl Woese developed was the 3 biggest groupings of life that he named the 3 main domains of life. And those 3 domains are bacteria. Let me get out my laser pointer. Bacteria, archaea, and eukarya. And this principle works simply the same way that a paternity test can work, or 23andMe. This is called a phylogenetic tree of life. Think of it as one giant parental test. Now, the reason why this is so important to microbiology is because of what was discovered and what came about in all of this. We once had a system of 5 kingdoms, not 3 domains. And for a while there, we focused on what was biggest and what was observable to the naked eye and easily observable, or what was most closely related to us because we view the world in a very human centric manner. So we had this animals group right here, right here, um, taking up its whole kingdom, 1/5 of all the kingdoms. But if you zoom in here and you look at the animals where the star is, it's a very teeny small part of the tree of life. Actually, animals are extremely closely related to fungi and plants and something called slime molds as well. There's not much of a difference between them genetically. Okay. And additionally, all of these other wee little beasties that exist here of the microscopic world. But on the other side here, bacteria and archaea, here we see the microscopic world, and it's not just here, of course, it's also in the eukarya tree, but all of these other organisms here are microbes. This really through the scientific community off its kilter here. So we've talked about some of these before. We've talked about the methanogens we can find here in archaea. Okay, we've talked about cyanobacteria over here in bacteria. We've talked about gram positives as well. And at the base of this tree is what we call our universal common ancestor. These are the ancestors that would have united all of the different branches here. All the domains originally came from this. Okay. Now, as you can see, recent developments in our evolution were animals and fungi and plants way out here. Further back here, we all had a last universal common ancestor. If you abbreviate that or take the initials, it's LUCA or LUCA, close to the base of the branches. Between eukarya and archaea, you would find the closest organism we have to this branching point was the one we talked about in the last lecture, the Loki archaeum. Okay, so these unicellular archaeum that look so much like bacteria are actually more closely related to us humans than to bacteria. Scientists are very interested in these interfaces, and there are a lot of mysteries surrounding this tree, such as when did archaea and eukarya split, and what events caused it to occur. Additionally, when did the archaea eukarya lineage split from the bacterial lineage? Here. Checkpoint 1. Who definitely and finally disproved spontaneous generation? How did he do it? Okay, what was unique about his experiment amongst all the others that finally put a close to this question? That completes the wrap up of chapter 1. Let's now switch focus to chapter 2. In chapter 2, How We See the Invisible World, we'll focus on how scientists viewed microscopic organisms. The learning objectives for this chapter are understand the form and function of the parts of the microscope, how microscopy works, compare the different types of microscopes, how to observe microbes using a microscope, staining methods for identifying and differentiating microorganisms. We will mostly focus on light microscopy because that's enough to handle in itself, and it's the most common type of microscopy. In particular, we'll be looking at bright field, dark field, phase contrast, and fluorescence microscopy. Although there are many other types of light microscopy, electron microscopy is common and uses beams of electrons to look at very small objects, whether that's subcellular parts or things like viruses. Scan probe microscopy inserts a tiny probe into a specimen and then transmits an electrical current through the probe to map the structure of the specimen. But again, we'll focus on light microscopy here. This is called a compound light microscope because it uses visible light and because there are two lenses in it, two main lens types in the compound light microscope. The first and most obvious one is the ocular lens, right here at the top. And this is the eyepiece, which magnified or remagnifies the image formed by the objective lens. The objective lens is right here, which are midway down the microscope. They are the initial primary lens that magnifies the specimen. Look at figure B, which will kind of give an overview of how the light travels through the microscope. It starts at the bottom. And then through the specimen and into your eyes. So if we look at the bottom, we have the illuminator. This is the origin of the light source, usually some sort of a light bulb down there. Old microscopes used to just have a mirror that reflected, ambient light in the room up through there. So the light will then travel from the light source up through the microscope through a condenser lens, which focus the rays of lights into a narrow beam that crosses the specimen. Then these light waves pass and cross each other and go to the objective lens right in the middle there. Okay, right here. And notice how they cross through where the specimen's located. So that's where it gets focused with the condenser lens. The specimen being analyzed is laid on this stage here, and you can see the stage a little bit better over here. And then the light travels up. It gets what's called refracted, back, which just means it gets curved back and forth into a prism here. Actually, it's more like this is actually reflection in this prism. My apologies. Reflection is being bounced off, refraction is being curved. And eventually it is straightened out and comes through these ocular lenses right here. And through all of this, the image actually gets inverted. So you can see the light actually crosses each other. And when one image ends up in the other eye, and the other side ends up on the opposing side. So you get sort of this mirror image that occurs. Okay, this can be a bit confusing because that means when you try to navigate the specimen by moving it around on the stage, moving the stage left and right, you actually go in the opposite direction than what you're seeing through the ocular lenses. So it's a little bit discombobulating when you're working with a microscope. So now that we've done that, let's list the different parts of the anatomy of the microscope. Again, let's start here on figure B, which shows the path of the light at the beginning. We have the base with the source of the illumination here, which is called the illuminator, the condenser lenses, which focuses the light onto the specimen. The specimen goes here at the stage, right, where it is placed. The objective lenses are above that and they magnify the image. We have the body tube above them, and then the prism inside that redirects the light towards the ocular lenses, the reflection. Let's go back now and look at figure A, which is a realistic picture of a microscope. And we'll show the different mechanisms and parts that are included in the microscope here. If we start at the bottom and work our way up, at the very bottom, you will find that there's some sort of an adjuster of the light intensity down there. And above that, you will have what are called focusing knobs. There will be one on this side and one on the other side. And so that allows each hand to be able to control the focusing. To change the focusing knobs move the stage up and down. So this will change how well things are focused. So instead of actually moving the magnifier, the magnifying lens, you're going to just be using the stage up and down, which will facilitate the same process there. Remember, the stage is what holds the specimen. We have two types of knobs. We have a coarse focusing knob, and that's a larger knob, and we have a fine adjusting knob, which is this inner knob right here. And can you guess what those two knobs do? What's the difference between a fine focusing knob and a coarse focusing knob? The answer is the coarse focusing knob moves the stage quicker, and as a result, it allows you to move in and out of focus more quickly than the fine focusing knob. The fine focusing knob is for fine tuning. So we're dealing with a very small amounts of changes, which results in very big differences in the focus that you actually end up seeing. Remember, these microbes are very small, so we have to be able to barely move that stage when we want to really sharpen the picture. And the fine focusing knob allows you to fine tune whatever you can't do with the coarse focusing knob. Let's keep working our way up the microscope. Besides the illuminator source, above that is the iris diaphragm. The diaphragm controls the amount of light entering the condenser. The diaphragm is like how your eyes will dilate in order to control the amount of light that goes through your eyes. So when you first wake up, things might be very bright because you haven't adjusted your eyes to let in just a narrow amount of light. So this iris diaphragm can control the amount of light that enters the condenser. You typically want more light for a specimen that's thicker, or if there's more mass on that specimen, so that you get less of a dark image caused by its shadow. The condenser is right above it, and the condenser once again focuses the light onto the specimen. It doesn't focus the specimen again. That's the job of the focusing knobs that move the stage up and down. It just concentrates the light onto the specimen, as we saw in figure B here, with how the light gets directed right into the middle there on the specimen. Um, so again, the stage is where you put your slide, which is a piece of glass containing your specimen. It's hard to see in this image, but there's actually some sort of a specimen holder or clip that flips out and kind of holds that glass there snugly in place on the stage. Next are the objective lenses. Here we can see there are different lenses which have different magnifications. So you can increase the magnification level by switching the objective lens. These objective lenses are attached to what we call the nose piece. The nose piece is this black rim you see, and it holds all of the different objective lenses around in a circle so you can rotate between different magnifications. The order you find your specimen and then magnify it is crucial. First, you start from a very low magnification, then find your specimen through the microscope. Once it's found, you fine tune that specimen. And we're going to go over that here in the next slide. So, steps for observing a specimen. First, lower the stage using the coarse adjustment knob. This just makes sure that the stage isn't going to bump into any of the lenses here, and you have plenty of room to put your glass slide. Next, rotate the nose piece to the lowest magnifying objective lens. This is typically going to be a 4x or a 10x, depending on the microscope. Now place your specimen or your glass slide on the center of the stage so that it is directly in line of sight of the objective lens. Raise the stage using the coarse adjustment knob, looking through the ocular lens until the specimen becomes in focus. Switch to the next magnification setting and use the fine adjustment knob. From here on out, sorry for the typo, from here on out, always use the fine adjustment knobs. Repeat the prior step until you reach 60x objective lens, and then rotate the nose piece so that it is between 60x and 100x, and then add a couple drops of immersion oil or lens oil. Finally, complete the rotation to 100x and use the fine focus adjustment knobs to focus in on the specimen. Now, there's going to be a lot of tips and tricks for getting this done properly, which we're not going to be able to cover because this is an online lab, but those are kind of the basics. Here is a zoomed in image of the business end of the microscope. If we look, we can see we have the glass slide in the middle, and not depicted here would be the stage that holds the glass slide. So if we work from the bottom up, we have our light source coming in. The left and the rights are just designated red and blue so that you can keep track of them. They're not actually red light or blue light. Underneath, we have the iris, and the iris is a movable piece and it can shrink. These two walls come in, right here, the iris diaphragm, and shrink around it, or they can expand in order to increase the funnel of light that comes through here. Above the iris diaphragm is the condenser, which can be moved up and down in order to control the angle at which the light comes in here and focuses on your specimen. So you want a razor sharp focus of light that comes here onto your specimen. Again, here's a glass slide of the specimen right here. So if it's upside down and the specimen is on the bottom of the glass slide, then you're actually viewing this incorrectly. You're going to rub off the material, your specimen, and your microscope won't work properly. So always make sure that whatever you fix to that glass slide, it is on the top of that slide. Then we have some space that will occur naturally between the specimen and the objective lenses here. And we talked about the immersion oil here, which is going to be really important when looking at things at high magnification. So what do we mean when we say that a lens is 10x magnification? What that means is that it's 10 times the magnification in which you would see with the naked eye. Same with 40x or 100x. 100x is 100 times what you would see with the naked eye. When we are looking through a compound microscope, we need to keep in mind that there are actually two lenses that are both magnifying the view of the specimen. We have the objective lens, which is the first set of lenses here, and we have the ocular lens, which is the lens to the right. Take a moment to review what the magnification of the ocular lens should be. Hopefully you thought that it was 10x. Typically, ocular lenses are set to 10x magnification. So how do we figure out the total magnification? It's a very simple calculation where we take the objective lens power and we multiply it by the ocular lens power. So if you have an ocular and an objective lenses both set at 10x, then what is the total magnification? If you said 100 times magnification, you'd be correct because 10 times 10 equals 100. Checkpoint 2. If the ocular lens power is 10x and the microscope is set to the high power objective lens or 40x, what is the total magnification of the specimen? Finally, let's discuss resolution. Resolution is very important in making really good microscopic images. Resolution is the ability to distinguish between 2 points. So depending on what device you're using right now, you may or may not be able to tell the difference between the 2 periods I have circled in red. If you can, you have a higher resolution than someone who cannot distinguish 2 periods in that line. Ultimately, What this means is that the lower resolution you have, the fuzzier the image becomes, and the higher resolution, the sharper the image is. And there are two big factors that affect this. The first being wavelength of the light. Light rays can come in different wavelengths, and we'll show that here in the next slide. And the shorter the wavelength is, the more capable you will be able to determine if there's that one period there or two periods there. The second factor is the numerical aperture. We've already discussed magnification, which is the ability to increase the size over the real size of an object against the naked eye. Numerical aperture is the lens's physical ability to gather light. And we're not going to get into the mathematics of that, but the more light that it can gather, the better. So with numerical aperture, the higher the numerical aperture, the better the resolution. Let's return to wavelengths. What do we mean when we say the wavelength of light? Each type of light has certain features about it. So here's a cartoon depiction of a light wave, and we can see here that the waves have a crest or the highest point and a trough or the lowest point in the wave. The distance between the trough and the crests, from the top to the bottom, is called the amplitude. And that's how intense that beam of ray of light is. Kind of like with sound, the higher the intensity of sound, the louder it is. That doesn't really change the innate characteristic of the light or type of sound, just the volume or intensity. However, the wavelength is what's unique to each type of light, and that is the distance between one crest and the next crest, or equally the distance between one trough to the next trough. Now, we can't see an individual wavelength with our eyes, but we do have a specific way of identifying them. Different wavelengths of light appear to us as different colors. The wavelength actually makes a huge difference when magnifying an object. The shorter these waves, the higher resolution we can get, because these shorter waves can overlap less and blend with other light and have less blending with other light traveling in the same direction. The longer the waves, the more they overlap with one another and the more blurry an image appears. What we call light in our day to day conversations is really just the portion of light us humans can observe. Another name for light is electromagnetic particles, and it comes on a whole scale. As humans, we can only see what we call the visible light portion of the scale. In this scale, blue light is the shortest of the wavelengths. You can see just here, this area right here is blown up in blue or purple, even is the shortest. Um, so, by using bluer light or more purple light in illumination, the resolution can be mildly improved. This is a limitation of light microscopy, right around 380 nanometers of light. That's the wavelength here, measured in nanometers. The resolution will be no better than the visible light spectrum, or down to about 380 nanometers or 0.38 micrometers, is another way to say that, where our vision ends. Can you recall how small bacteria are? Somewhere around 1 to 2 micrometers. And again, 0.38 micrometers is the size of a far purple light there. And how small are viruses? Well, generally, 0.3 micrometers or smaller. Therefore, you can see that even our best magnification makes it pretty difficult to see much detail with bacteria, and nothing at all when it comes to the size of a virus. So when we observe images under the microscope, it's a much different ballgame than what you see blown up for our viewing pleasure on these slides. Our maximum magnification using bright field light microscopes in most labs will be about 100x for the objective and 10x for the ocular, meaning about 1,000 times. A 1,000 times magnifying bacteria, that is 1 micrometer, will appear to be the size of 2 periods in 12-point font size. So you should get comfortable identifying the bacterium shapes and phenotypes in these slides, because just imagine what the real world applications of microscopy will look like. I'd like to ask you to take a moment and just note where gamma rays, X rays, and ultraviolet rays are on this scale. We will discuss them in more detail when we cover methods of controlling microbial growth and mutations. You don't have to memorize all of the different nanometers for all of the lights here. That's not what's important here. Just a general concept of the ideas we're discussing here. Let's talk about the different types of light microscopes that are out there. The first one and the simplest one and the most common one is the bright field microscope. This uses visible light to illuminate a specimen. Imagine the light being filtered through the diaphragm, through to the condenser, and onto your specimen. The specimen is going to block out or shade some of the light, and anywhere there isn't a specimen present or no objects present on that glass slide, there will be no shading, and it will appear as a bright image in that place. So the specimen is actually a darker image surrounded by a bright background, or in other words, a bright field. The resolution limit here, again, if you're amazingly good with your resolution, is allegedly zero point 0.2 micrometers. Here we have a big paramecium that is about 100 times as wide as a bacterium, or more, actually. In practice, zero point 0.2 micrometers leaves us wanting a lot more and doesn't give us the level of information that's sometimes necessary. Dark field is naturally the next type of light microscope to discuss after bright field. Before going further, let's take a moment and I want you to think to yourself, why is this called a dark field? If you are looking at this image, what are you likely to see as bright? And what are you likely to see as dark in a dark field microscope? Just intuitively here. Dark field uses a special type of disc that blocks light from the center. You can see it right here from reaching the specimen. Okay. This means that the only light that makes it to the objective lens is light that gets refracted, bent as it passes through the specimen, back towards the center. This light right here as it passes through the specimen. Dark field microscopy uses a special condenser to block light from entering the objective lens. It does so with a small disc, as we can see close to the light source. It's about 1 centimeter wide and blocks most of the light. So you should have guessed that you believe that the field or the background is going to be dark and the specimen is what's going to appear illuminated. And that has to do with that little disc. What happens is only the light on the edges is condensed back to the specimen and it hones in the middle of the sample, which is then reflected or bounced off the specimen and enters the objective lens. And that's what you end up seeing. As a result, the image is inverted and only the specimen is illuminated. This is really useful for visualizing organisms that can't be seen using bright field microscopy. Typically, those are organisms that don't stain very well. So, this could be a live organism you don't want to destroy in the staining process, or just organisms that you don't end up using a stain for one reason or another. Brightening up only the specimen helps to highlight it when staining cannot be done. Here is an example of a bright field microscopy image, and we can see this is a paramecium. If we go ahead and take a look at this scale bar, we can see the 25 micrometers in scale. So this is probably about 6 to 9 times that scale bar size. Excuse me. So this would confirm that it's about the correct size for a paramecium, around 200-ish micrometers. Something really beautiful about dark field microscopy is that we can see different parts of what is called the ultrastructure of the cell or the inner parts of the cell. So there's some organelles and things that we are seeing inside of the cell that you would not be able to see with bright field. And excuse me, this is actually a dark field that we're seeing in this image. Phase contrast is a very complicated form of microscopy. Two types of light that enter the objective lens. There's direct light from the light source, and then there's also the reflected or diffracted light from the specimen itself. Generally, there is not any need to fix or stain with this sort of phase contrast microscopy, and it provides these very rich, deeper visualizations of a specimen, and it allows for much better visualization of internal cellular structures. So you can see that all of these internal structures have much more contrast and tend to pop out a lot better than bright or dark filled. We're not going to get into too much detail about how these phase contrast microscopes work, but suffice to say that it uses physical properties of waves to add light waves together, and therefore amplifying their intensity. And the image creates a much deeper and richer visualization. Let's take a closer look at how this works. Imagine waves in an ocean. And if two separate waves are moving in the same direction and end up coming together and overlapping, depending on how the waves match up, they can cancel each other out or make each other stronger. In the image above, the waves are both going up and down at the same time as they come together. They reinforce each other and lead to what is called constructive interference. If one wave is going up and the other is going down at the same time, they cancel each other out, and this is called destructive interference. The same can be applied to light waves. Remember that light refracts as it passes through an object? In typical bright field, this refracted light leads to shadowing. And that's because wherever the light gets refracted out, it doesn't make it to the objective lens, and so it's darker in that area. We call that a shadow. In phase contrast, that light is carefully added back in. It's reflected back into the image perfectly so that it either forms constructive or deconstructive interference. We won't bother going into any more depth on the mechanism. You need to just be aware of the basic function of constructive and deconstructive interference, and this is what causes the richness of the image. Another fun type of microscopy is fluorescence microscopy. And this again uses visible light, sometimes even all the way to those really short wavelengths, which are called ultraviolet. So that would be a little smaller than what we are able to pick up with our own eyes. And this ultraviolet light is dangerous and harmful, so there is an extra precaution which we'll discuss later that's used to filter out this light for the end imagery. And it makes these very bright images. And the way it does this is it uses either naturally fluorescent organisms or organisms that are stained with what we call fluorochromes. These are fluorescent dyes that attach to the structures of the cell. In our example, ultraviolet light will stimulate the fluorochromes and cause them to get excited in the specimen. And then they release their own light, similar to how fluorochromes found in glow in the dark toys work. You charge them up in the light and then you turn off the light and they give off their own glow. Fluorescence microscopy has specialty purposes. Certain species of bacteria can be effectively stained and detected with this technique. This image is from the OpenStax textbook and is a fluorescent stain used to visualize larvae of Schistosoma mansoni, which is a parasitic worm that causes intestinal disease, commonly in the tropics. And importantly here, a filter is going to be used after the specimen that blocks all other light except for the color of the fluorescent marker. So whatever light that is being fluoresced off. In this example, it's the green light. And if it's excited with a UV light, that ultraviolet light gets filtered out so it doesn't harm the eyes or confuse the person who's trying to view the specimen by having all that other interference light. And so the samples are going to appear in bright color against a darker background. So again, a specific wavelength of light is shined on the specimen. The specimen's chlorochromes, or its natural fluorescent chemicals, are excited and release a different wavelength of light. The fluorescent microscope has a filter on it before the light reaches the ocular lens, and this filters out all but the light emitted from the fluorochrome. So, in our case of ultraviolet light being the excitation light, none of that makes it through and interferes with your ability to view the fluorescing light or causes any eye damage. One example of a fluorescing protein found in nature is what we call green fluorescent protein, or GFP for short. It's a very common fluorescent marker. It's coded for by a very small gene of about 300 base pairs in length, and which in terms of genes is about half the size of the average gene. It's often used as a tag for bacterium. So in the image you see off to the right, I GFP tagged a cyanobacterium. Essentially, I put this gene, genetically engineered this organism to carry the GFP gene. I also had another gene that I inserted in this organism at the same time. So I could tell if the organism had the green fluorescent, had the gene that I was trying to put into it, if it fluoresced green, because they both came together in the same genetic modification. So it's often used as a tag for bacterium, and it fluoresces a brilliant green color, hence its name. This is the GFP protein present inside of the cell. So this is the image that was first exposed to UV light, and then the specimen itself gave off this green fluorescent color, and then a filter was placed between it and the ocular lens, which blocked out the UV light but allowed the green light to pass as a consequence. So we're only seeing the green color. And we actually do a mini version of this experiment in our primer design and cloning lab. This image right here of Candidatus prometheoarchaeum syntrophicum is closely related to archaea, bacteria, and eukarya. So it's very low on that phylogenetic tree. And what this means is that we are really close to the base of the phylogenetic tree of life, where life diverges into the three domains. There's actually a culture of many organisms in this sample here, all working together in symbiosis. And the presence of other organisms and each one of those organisms are very hard to see because they're little bacterium or archaeum clustered together. But by using fluorescence microscopy, we can pick up fluorescent signals from different bacterium, and we can use a computer software program to count them and generate the populations of bacterium that exist in that culture. As you can see in these pie charts, it lists the different organisms living together in each one of these images. In this way, scientists were able to see what microbes were working with each other or in a symbiotic relationship in this community by their cohabitation with each other. Without the color, we would do a poor job at best at identifying which microbes each of these small little dots were. But since each has their own unique fluorescent signature, it becomes possible to process and quantify their numbers. That brings us to our discussion of how samples of microbes can be prepared for light microscopy. There are two things your mother never wanted you to do, and that was staining and smearing. But in microbiology, we do a lot of staining and smearing. So, what is staining and smearing? Well, staining is a way of coloring a specimen with specific dye that emphasizes certain structures. However, before staining a specimen must be attached to the slide. These specimens are almost always a culture of many microbes. The specific term for these broth cultures is called liquid media. And by creating a thin layer of this culture on the glass slide called a smear and making sure it is well fixed to the slide so it doesn't come loose during processing and examination under the microscope, we can then begin the process of staining. The steps of staining begin at the top of this image. It can be done from liquid cultures left on the left side or cultures grown on solid nutrients like a petri dish on the right side. The culture is spread with an inoculating loop from liquid media by dipping the loop in the liquid broth. And this loop is much like a bubble wand. Liquid forms a film in the loop, and we can be and it can be transferred. And again, here's the loop right here. And it can be transferred to the slide and smeared around in the small circle, like so. A quick examination must show a very, very faint smear after you've deposited your smear there. And we must avoid having too thick of a layer because we don't want the microbes laying on top of each other, obscuring a clear view of each of them. For solid media like a petri dish, in other words, microbes grown on one of those plates, we have shown when we discussed how penicillin mold prevented bacteria from colonizing the area and led to the development of antibiotics. An inoculating needle is used, not a loop. See right here is a needle to pick up a small amount of sample and smear it onto the glass slide. The slide is allowed to dry at room temperature for several minutes so that it no longer has any moisture visibly present. And finally, the slide is fixed by passing through a flame quickly a few times. Sometimes labs do not have flames available, and an electric heating element is used, called an incinerator, which is essentially just a little ceramic heating element, and that's not quite as aggressive as an open flame. So you'll hold it against the heating element for, maybe 20 seconds, 30 seconds, just enough to kind of warm the glass up until it is too hot to want to touch, but not any more so that you don't overcook the organisms and destroy them beyond recognition. You want to use a clothespin to do this, or a pair of pliers or something so that you don't burn your fingers. And you also want to make sure that if you look under the microscope and you don't see any bacteria, that might have been for two reasons. You did this for too long and you destroyed your cells, or you did not fix for long enough or with enough heat. And during the staining process, you washed away the cells because they weren't fixed. So it takes a bit of troubleshooting to get the hang of it. And admittedly, it is a bit tricky. Stains are composed of different chemicals. Stains are salts. A salt is a chemical compound made up of a positive and a negative group. So stains are composed of a positive and negative group called an ion. Here you might need to do a little bit of a review if you don't remember what a positive and a negative ion are. Essentially, a negative ion has electrons and an excess of electrons versus the number of protons. Protons are positively charged, electrons are negatively charged, and that just makes the atoms in that molecule more negatively charged. A positive ion has a fewer number of electrons than it should compared to the protons, so that makes it positively charged because the protons outnumber the neutrons. In other words, it is lacking in those electrons which have a negative charge to them. So to summarize, either the positive or negative ion is a chromophore here. The chromophore is a part of the stain that is colored. In basic dyes, the colored portion of that salt resides on the positive ion. In acidic dyes, the portion that is colored is the negative ion. I repeat, in basic dyes, the colored portion of the salt resides on the positive ion. In acidic dyes, the portion that is colored is the negative ion. Bacteria have negatively charged surfaces or cell walls. Therefore, will basic or acidic dyes stick to bacteria? Well, if you're familiar with how positive and negatives attract, you should have guessed that positive ions will be attracted to their negative cell walls, and negative ions are repelled by their negative cell walls. So, basic dyes will color the bacterium, and acidic dyes will color the background and avoid and be repelled by the bacterium. Sometimes you want to stain the background, and this gets a little confusing because a background stain is called a negative stain, and staining the bacterium is called a positive stain. You can remember this because a positive stain has a positive ion and it is a basic dye. A negative stain has a negative ion, chromophore, and it's an acidic dye. The reason why acidic dyes don't stain the bacterium is again because they're both negatively charged. Therefore, you will have some residual negative ion chromophores that stick to the glass, but you should have clear zones where the bacteria is because they are repelled by the negative charges of the bacterium. There are three different kinds of staining techniques that we can do. The first and most simplest one is simple staining. It uses just a single basic dye. And remember, basic dyes are going to attach to the outside of the cells. This highlights the entire microorganism and allows us to visualize the cell shape and sometimes internal structures. In the example you can see, methylene blue provides a simple stain of human cheek cells. Simple staining is simply a single basic dye. As you can see, the cells are colored and the background is not. This is good for staining cells you already know what they are and need to visualize them for analysis. Differential staining involves identifying an organism from possibly other organisms. Differential stains tend to be much more complex. They involve multiple dyes, and what makes it a differential stain is that it is used to distinguish between two types of bacteria or cells, the most common of which are Gram stains and acid-fast stains. In the example, imagine you can see this is a Gram stain of Lactobacillus crispatus, and it's purple, so it is what we call a gram-positive stain. Again, that should be a capitalized gram, and we'll discuss in a moment here why purple makes it gram-positive. The Gram stain is by far the most common stain used in microbiology. You will need to memorize all of the steps for all of the following staining techniques. The chemical agents change between differential stains, but there are many similarities. Here are the general steps in the Gram stain. Step number 1 is the addition of the primary stain. In the Gram stain, it is crystal violet. Crystal violet dyes all of the cells a purple color, regardless of them being gram positive or gram negative. In this first step, so any cell present, you can see it's going to be purple after step 1. Okay. And step number 2 is the addition of a mordant. A mordant is an ion that binds to that dye and helps it to hold itself in place better. Sometimes we might even use heat as a mordant, as in the acid-fast stain that will come later. In this example, it's a chemical mordant called iodine that creates what we call the crystal violet iodine complex. The next step in this process is to get rid of the primary stain from one type of cell so that we can apply our contrasting stain to those cells. Here we have an alcohol wash. We apply alcohol for just a few seconds in order to cause the decolorizing to occur. Not anymore, or else you could decolorize all of the cells, not just the cells that you're trying to counterstain. And those that are decolorized are going to be the gram negatives. They will lose their dye readily. However, those that are gram positives will retain the dye a lot more strongly. So at this point, we should have our gram positives as purple, and the gram negatives will be colorless. And then lastly is the secondary stain that you see here. Now, the secondary stain is what's going to dye the gram-negative bacterium pink, and it's the application of a stain called safranin. And it's going to stain everything pink except for the organisms that are already purple. It's not going to change the color of those at all because the crystal violet iodine complex is going to be overwhelmingly more strong than that pink color itself. And so if we look at this image here, our actual example, we'll see we have two different bacterium. That's obvious because one of them are bacillus shaped and one of them are coccus shaped. But in this image, we can determine which one is gram positive. Can you? You should have said the coccus bacteria because it looks kind of blue in this image. Although crystal violet is more of a bluish purple color, Then you have the gram negatives, and those are the rods. Well, why do some bacteria stain gram positive and others stain gram negative, you might ask? This is because the bacteria with a very thick layer of peptidoglycan in their cell walls retain the crystal violet during the decolorization step. However, the bacteria with a thin layer of peptidoglycan in their cell wall easily and readily gives up that color during the decolorization step. So this results in gram positives retaining their primary stain, and they are purple in color, and they're called therefore gram positive. Gram invented this stain, so that's where they get the name from. The gram-negative bacterium lose the primary stain during washing and then take up the counter stain and therefore are pink in color. Obviously, this allows an examiner to differentiate between two major groups of organisms or to start to classify what organism they might have. And this is why it's a differential stain. It allows an examiner to diagnose a disease or biological contaminant or an environmental sample. Also, we'll see just how important it can be to know the type of cell wall a bacterium has. The cell wall is the interface of the cell to the external environment, and the thickness of the cell wall has consequences for what type of antibiotic is effective against the bacterial species, as certain antibiotics work for gram-positive species and not for gram-negative ones. And I forgot to show you here the components here of the thick peptidoglycan layer. They retain the primary stain. They're purple in color and called gram-positive. And the opposite is true for gram-negative bacterium. The next stain we're going to discuss are acid-fast stains. Acid-fast stains work on two types of bacterium that have waxy material in their cell walls. This includes bacteria from the genus Mycobacterium, which are bacterium such as M. tuberculosis, which causes tuberculosis, and M. leprae, which causes leprosy, and also Nocardia, some of which are non-pathogenic and others are weakly pathogenic to those who are immunocompromised. So this stain is very important for screening for these organisms in the medical field. This is how the acid-fast stain is conducted. Once again, the primary stain is added first. This primary stain is a very common stain used called carbonyl fusion, and it is applied to a fixed smear and colors all cells pink. Then we're using heat as a mordant this time. So the slide is gently heated. Here we have a little heater and a water bath that the slide sits over. So just so it doesn't get too dried out in the mordant process. Then we have the decolorizer. Okay, and the decolorizer, of course, we want to be able to differentiate. So we need to get rid of the primary stain where it doesn't belong. So we use an acid alcohol decolorizer. And then finally, we add our counter stain in an acid-fast stain. The counter stain is methylene blue. And this stain will stain all cells without a waxy cell wall that were decolorized in the prior stain. A couple things to note here. This is kind of the opposite of a gram stain. Remember that a gram stain, the gram positives were purple and the gram negatives were pink. Well, in this example, carbonyl fusion is pink and methylene blue was purple or bluish purple. So the counter stain is actually the purple one and the primary is the pink. Another thing to note is, why do we use that heat? Well, we use that heat because these waxy cell walls are very difficult to penetrate. And there's a good reason for that. It's a defense mechanism for the mycobacterium. And so once again, those cells that do not have that waxy cell wall, they readily give up the primary stain to that decolorizer. So it's kind of the same as a gram positive in this mechanism of being able to retain the primary stain better than acid-fast negative bacteria. Checkpoint 3. Interpret the gram stain below. Is the circled bacteria gram positive or gram negative? Checkpoint 4. Interpret the acid-fast stain below. Which letter, A or B, could be M. tuberculosis? Some microbes contain a protective coating called a capsule. Capsules are important to many pathogenic bacteria because it protects them from harm by the immune system. This capsule can indicate virulence, which is the degree to which a pathogen is disease causing. These capsules do not take up um, most dyes very well and appear as an unstained halo around the cell when stained with India ink or negrosin. This is a type of special staining that is done and is referred to as negative staining because the background is stained and the organ, the cell wall itself, is not stained. The next one is endospore staining. Endospores are another structure by the cells. You can think of endospores as bomb shelters where bacteria can hunker down and survive rough times. These bomb shelters are consequently also very difficult to stain, but it can be done using a lot of heat and time to weaken the structure and allow the dye to absorb. Endospores arise in certain bacteria under stressful environmental conditions. The staining requires the Schaefer-Fulton method. The primary stain is malachite green, and it uses a heat, and decolorizer is just water, and a counterstain of safranin. The consequence of which, as you can see in this image, is that the endospore sustains a green color from the malachite green, and then the counterstain is the pink. So those endospores hold on to the primary stain really well and appear to be a greenish color. Microbiologists use carbon fusion, again, to layer a thick dye that can be visualized. Um, in our last discussion is flagella staining. So flagella are these little appendages that are attached to the outside of microbes, some that typically help things like motility. Most bacteria are only 1 to 2 micrometers thick, so these thin appendages are extremely hard to see with the resolution of a microscope. An unstained flagella is extremely small and hard, if not impossible, to see with a light microscope. But we can use a carbofusion and a mordant that decolorizes the flagella that coats the flagella with a thick enough coat to be visualized under light microscopy. Checkpoint 5. Place the steps of a differential stain in the correct order: mordant, decolorizer, counterstain, and primary stain. And finally, checkpoint 6. Would endospore staining be classified as a simple stain or differential stain, and why? And that concludes the end of chapter 2. Please join me for chapter 3, where we'll discuss the different parts of the cell.